welcome you all in CEC-UGC Gurukul live lecture. Learner, in previous part we had discussed about the nucleic acid and DNA, what is DNA and what are nucleotides. As I have told you, the nucleotides is composed of 3 units. One is sugar unit, other is a phosphate unit, and third one is a base, and bases are attached to anomeric carbon of the sugars, and on the basis of sugar we can give the RNA name RNA and DNA. So this is the structure on this screen. On your screen you can see the double helical structure of DNA. As I told you, the DNA is the most complex structure and it is a macromolecule, macrobiomolecules, and having a monomer unit, that is a nucleotide. So it is this structure given by, proposed by Watson and Crick in 1953, and they have concluded the secondary structure of nucleic acid. So this double helical structure is the secondary structure of nucleic acid, and here the sugar phosphate backbone on the outside. As I told you, the sugar anchors the phosphate formation of phosphate bond, and that sugar phosphate bond sits on outside of the DNA, or we can say it is a backbone on the outside and bases are present inside the double helical structure, and both the chain of nucleotides are held by hydrogen bonding between the base on strand, bases on other side. So one base of one strand held by hydrogen bonded for other hydrogen bonded of other strand base, and distance between two strand is relatively constant. And it's very important point to remember here, in both the strand the number of purine bases must pair with the pyrimidine bases. So purine bases always paired with the pyrimidine bases. So if the larger purine bases is paired, what happen if larger purine bases are paired in the DNA strand? So here the strand would bulge, and if smaller pyrimidine bases facial the strand, it would have to come into bring two pyrimidine too close enough to form a hydrogen bonds. So DNA is composed of nucleic acid chain and running anti-parallel to one another. This is very important characteristic of DNA. Both the strands running anti-parallel to each other. So side by side with the 5 end, as I told in the previous slide, 5 end on one chain and next to 3 end of other chain. So on your screen you can see here 5 end on the one end and 3 end on the other end. So the two chains are twisted to form double helical structure. And what is the role of base pairs? Learner, base pairs are very essential for replication and forming a helical structure of DNA. Bases are part of DNA that store information and give DNA the ability to encode phenotype, a person's visible traits. Now we will discuss the nomenclature of nucleotides. So on the basis of base present in the nucleotides, we will discuss here how can we give them name. So first base, purine base. If the purine base is adenine and strand is nucleoside, nucleosides, always remember nucleosides contain base plus sugar, right? Only base plus sugar, no phosphate bond, no phosphate group is present in the nucleoside. So if nucleoside having adenine base which is attached to the C1 anomeric carbon atom, we call deoxyadenosine. And if the sugar is deoxyribose sugar. Similarly, if there is a nucleotide, we called it deoxyadenylic acid or deoxyadenosine monophosphate. Next, if the base is purine base is guanine base and there is nucleoside and the sugar is deoxyribose sugar, we called it deoxyguanosine. And if there is a nucleotide, we call deoxyguanol-like or deoxymonophosphate guanol-like. Similarly, if the base is cytosine base, so these are the purine bases, and if there is a nucleoside, we called it deoxyribose cytidine, and if this is the nucleotide, we called it deoxycytidilic acid. And if the base is thymine base and if the strand is having nucleoside, we call sugar is deoxyribose sugar, we call deoxythymidine. Similarly, in nucleotide, we call deoxythymidilic acid. And if there is a uracil base, as I told you, the RNA contain uracil base, and if there is a ribose sugar, because always RNA have ribose sugar. So in this case, the nucleoside, we called it uridine, right? And in nucleotide, it is called uridylic acid or uridine. So this is the structure. Here, in nomenclature you can see here, this is a base and sugar. So sugar H, I have written here in the bracket OH. It means that if there is H, it is deoxy, and if there is OH, this is oxy. So it is deoxythymidine. This thymine base is present here, and also called ribofuranosyl thymine. And in abridged structural formula, the nucleoside can be represented in this way. On the top carbon, you have to write thymine or any base, and there is a two horizontal bond which represent the OH group. Similarly, this is the abridged formula where there is a base which is guanosine. So it is the abridged formula, or in a short form we can write the nucleotide structure. It's very important and it's very easy to learn how we have to write the nucleotide structure. So it is the simplest way in abridged form. So another structure you can see, there is a adenosine base. So in adenosine you have to write A on the top and the two horizontal line which represent the hydroxyl group. Similarly, it is the structure of nucleoside. This is the structure of nucleoside. Why this is structure of nucleoside? As I told you, at 5 carbon atom nucleoside having CH2OH group, but in nucleotide that OH group, that H is replaced by phosphate group. So it is very easy, you can understand and you can identify which is the nucleoside, which is the nucleotide. So it is the structure of nucleotide and the base is present here, cytidine, and it is abridged structure formula. So in abridged structure formula, sugar represent in the vertical line. So vertical line, there is a two line on your screen you can see here. One is vertical line, two are horizontal line. So always remember, vertical line represent the sugar while horizontal line represent the hydroxyl group. One is second carbon atom and other is at the 5 carbon, second carbon, third carbon, and if there is a nucleoside, the 5 carbon have CH2OH group, and if there is a nucleotide, the 5 carbon have CH2OP4, right. So this is the abridged structural formula. By these abridged structure formula, we can easily write the very complex structure of nucleotide and nucleoside in a simple manner. So here in the second structure you can see the second carbon atom H is absent, so it will be deoxy, right. If OH is absent at second carbon atom, it means that the sugar is deoxy sugars. Learners, as I told you how the double helical structure of DNA base pairing takes place. So on your screen you can see American biochemist Erwin Chargaff. So Erwin Chargaff proposed a formula which is called Chargaff rules. On the basis of Chargaff rule, he described that his research described that the number of bases in DNA equals to the number of, like if there is a adenine, so adenine is equal to thymine and thymine is equal to guanine and guanine is equal to cytosine. So you can see on your screen A equal to T and G is equal to C. So pairing always takes place on the base of Chargaff rule, who proposed the number of, like here, purines which is having adenine and guanine, pyrimidine which are cytosine and thymine. So A should equal to T in a base pair in a DNA. A is adenine. Number of adenine base is equal to the number of thymine base. Similarly, number of guanine base should be equal to the number of cytosine base. So in DNA, the base exists into 1:1 ratio, and it is the basis of base pairing in DNA molecules. So base pairing are the fundamental units of double stranded nucleic acid, where the number of base are same. So adenine always pair with thymine and guanine always pair with cytosine, and these in two strands, we are called complementary strand, where adenine is one strand and thymine in opposite strand are linked together or paired together through the hydrogen bonding. So it is dehydration reaction takes place and both the bases present in both strand of DNA joined together. So base pairing is dictated by hydrogen bonding, as base exists always in keto form. Adenine forms two hydrogen bonds. So it's very important, because you can ask how many bonds can be formed in the double helical structure of DNA. Always remember, adenine forms two hydrogen bonds with thymine, and similarly the another bonds, the cytosine can form three bonds, hydrogen bond with guanines. So the DNA strand here in the structure you can see here, this is a clear structure where guanine and cytosine. So it is a part of strand, just written here guanine base and cytosine base. So this is the interior part of the DNA. So here you can see how the hydrogen bonding takes place. So the carbonyl group of guanine is joined with NH2 group of cytosine. So in this way the guanine form three hydrogen bonds with cytosine, and hydrogen bonds holding the bases together are all about the same length. So it is the interior part of double helical structure where the base pairing takes place, and during the base pairing all the bases should have equal number and joined together by hydrogen bonding. Now what is the base sequences? Learners, by convention, base sequences is a portion of nucleic acid which are written in 5 to 3 direction. 5 represent carbon atom which is having a CH2PO4 group. Similarly 3 is also having carbon atom of other nucleotide. So 5 end is on the left side and 3 is on the right side always. So it is a base sequence. On your screen you can see a base sequence. 5 end is on the left and third and 3 carbon atom is on the right. So AG, GA, GC, CA, TG, AG, all these are bases: adenosine, guanine, adenosine, guanosine, cytosine. So this is a base sequence. So 5 and 3 designation refers to the number of carbon atom in deoxyribose sugar molecule to which a phosphate group is bonded. And DNA is always read in the 5 to 3 direction. So it is very important point. Always when we have to count the base pairing, we have always read DNA into 5 to 3 directions. So DNA strands are not linear, but it is twisted into a helix which is around into the common axis. The base pairs are planar and parallel to each other on inside of the helix. So this is the structure of DNA molecule, DNA nucleotide, where you can see the nitrogenous base, all the nitrogenous base, purines and pyrimides inside of the helix. Sugar phosphate, which is the backbone of the nucleic acid, is outside of the helix, and the base pairs adenine to guanosine. So base pairing takes place through the hydrogen bonding. So on your screen you can see here the sugar phosphate is a backbone of DNA, and it is on the outside of the DNA molecule. DNA is nucleic acid structure, and adenosines pairing with the thymines, while guanosine is pairing with the cytosine. So always remember, the base pairing takes place with adenosine will not pair with the guanosine. Always adenosine pair with the thymine with 2 hydrogen bond, cytosine paired with the guanosine with 3 hydrogen bond. So the 2 strands are anti-parallel, they run in the opposite directions. So it is the double helical structure, and it resembles like a ladder, and the base pairs are rugs twisted around an axis running down through its rugs. So the phosphate group or phosphate sugar backbone is wrapped around the base, and the OH group of phosphate has pKa value about 2. So it is in basic form, which is a negatively charged at physiological pH. The negatively charged backbone repels the nucleophiles, so they can preventing cleavage of phosphodiester bonds. Unlike DNA, RNA is easily cleaved because the OH, which is present at the 2 carbon atom of ribose sugar, can act as a nucleophile that cleave the strand, and this explains why the OH group is absent in DNA. So it is the main reason why the only 3rd carbon atom have OH group in the DNA, because it's cleaved easily. So these are the some important characteristics. To preserve the genetic information, DNA must remain intact throughout the life span of cell. Cleavage of DNA would have disastrous consequences for all and for our life itself. In RNA, the OH group, which is present on the pentose sugar, act as an intermolecular nucleophilic catalyst, and it has been estimated that RNA is hydrolyzed 3 billion times faster than DNA, and hydrogen bonding between the base pairs is just one of the forces. It's very important to understand why the base pairing is important in DNA in double helical structure of DNA, because base pairing is the forces which holds the two DNA strands double helical stacks together. The bases are planar aromatic molecules that stack on the top of the other, and each pair is slightly rotated with respect to the next pair, like a particularly spread out hand of cards, just like that. There are favorable van der Waals forces of interactions between the mutually induced dipoles of adjacent pair of bases, and these interactions called stacking interactions. These van der Waals forces interactions between the bases are called stacking interactions. And what are the role of stacking interactions? Stacking interactions are weak attractive forces, but when added together they contribute significantly to the stability of double helical structure. So that is why DNA is the most stable structure, and clarify all the characteristic of genes, and also responsible for the passing of traits of from parents to their offsprings. So the stacking interactions are the strongest between purines and weakest between two pyrimidines. So that's why the base pairing takes place between two purines and pyrimidine bases. Confining the bases to the inside of helix also stabilize the helix, because it reduces the surface area of relatively non-polar residue exposed to water. This increases the entropy of surrounding water molecule and makes the DNA stronger and stable molecules. So it is if we talk about the helical structure of DNA, naturally occurring DNA molecule can exist three different type of helical form. The B helix is, on your screen you can see A helical form, B helical form and Z helical forms. So the B helix is the predominant form in aqueous solution, while A helix is the predominant in non-polar solution. The B and A helical forms both are right-handed forms. So it is the B helical structure. It's very important to understand here. Nearly all the DNA in living organism, also in plants, found in B helical structure. So all the living organisms have B helical, double helical structure, which is in B form. The dimension of B form is 0.34 nanometer between bond pairs and 3.4 nanometer per turn, and about 10 base pairs per turn, which is 1.9 nanometer is diameters. So B DNA has 10 base pairs turns and a 3.4 angstrom rise in B form DNA. Major groove is wider than the minor groove. So you can see there is a major and minor grooves. On your screen you can see how I have represented the major and minor groove. So the major grooves are wider mirror grooves. Proteins, which is found in the proteins, and other molecules combined together on the basis of grooves. So the hydrogen bonding properties of the functional group facing into each other grooves determine what kind of molecule will bind to the grooves. So here there is a surface polymers you can see here, and the downwards there is a minor grooves, and the upwards you can see is a major groups between the two base pairs Now we talk about the A form, A helical structure of DNA. So A form of nucleic acid and Z form of nucleic acid DNA structure. So A DNA has, it's very important, A form of DNA has 11 base pairs per turn and 2.3 angstrom rise. A thicker right-handed duplex with a shorter distance between base pair has described for RNA DNA duplex and RNA RNA duplexes, which is the A form of the nucleic acid. A helix is shorter and about 3% broader than the B helix, which is broader and shorter than the Z helical structure of DNA. Learners, Z helix is the left-handed helical structure. It occurs in regions where there is a high content of guanine and cytosine base pair, while Z DNA has 12 base pairs per turn and 3 angstrom rise. Z DNA is formed by stretches of alternating purines and pyrimidines like guanine, cytosine, guanine, cytosine, guanine, cytosine. So these are the alternating bases, purines then pyrimidine, respectively in negatively super-coated DNA. So the G and C multi-conditions, multi-nucleotides or polynucleotides are different in configuration, leading to zigzag patterns, and 3 carbon atom which is present in endo conformation, and guanine, if there is a guanine base, which is present in the syn conformation. So this is the base sequence. It is called palindromes. So what is the palindromes? And you can see on your screen, it is a base sequence which is present in the DNA. It is called palindromes. So learners, palindromic sequences are referred to as the sequences of nucleotide in DNA duplex or in RNA. It consists of 2 identical or similar inverted repeats. Repeats of what? Repeats of base pairs which are either adjacent to one another or separated by a separate regions. So you on your screen you can see palindrome base sequence or their mirror image. So what is the difference between A form and B form of nucleic acid? The difference is in the configuration of deoxyribose ring. It is C2 carbon atom which is endo conformation in A form, and the placement of base pair with the duplex is also different. In B form of DNA, the base pairs are almost centered over the helical axis. In A form, the base pairs displaced away from the center axis and closer to the major grooves. As a result, it is a ribbon-like helical structure with a more open cylindrical core in A form. So in the B form, it is the right-handed, A form also right-handed, but in Z form, right-handed helical sense. Similarly, B form have 10 base pairs, A form have 11 base pairs, Z form have 12 base pairs, and the vertical rise per turn in A form and A form, B form and Z form are different, which are you can see on your screen. Similarly, the rotation per base pair is same in A form and B form, while in Z form it is minus 30 degree. Helical diameter in all the form of helical DNA A, B and Z is 19 angstrom degree. So 3 major DNA which are having double stranded converted by interactions between complementary base pairs, these are terms A form, B form and Z form of DNA. If we talk about the conformation, all A in if there is a glycosidic bond with pyrimidines, so all forms have anti-conformation, but if glycoside bond with purines, so all the form have anti-conformation except Z form. Z form of DNA have syn conformations. So, base in DNA molecules repeat of specific sequence, this is palindromic repeats in DNA is a sequence of nucleotides, and it occurs when complementary strand of DNA and it is read thus in same direction as 5 pairing on the top and with the 3 pairing end. In we are talking about the DNA duplex or in RNA. So, example you can see here, I have represented a 5 to 3. So, this is the nucleotide sequence of one strand, it would be the same as reserved complementary strand. So, nucleotides always pairs in the same way, adenine, thymine in DNA or uracil in RNA, while cytosine with guanine which is a single stranded nucleotide sequence is called palindrome if it is equal to its reverse complements, and this palindromic morphids are found in most genomes or sets of generic instructions. Learners, human chromosomes, because human have 2 types of chromosomes, and on the basis of that chromosomes sex are determined. So, human have 2 types of chromosomes, X chromosomes and Y chromosomes, and they are arranged as a palindromes way. It allows the chromosomes to repair itself by bonding over at the middle, one side is damaged. So, biosynthesis it's very important. Biosynthesis of DNA. Learners, biosynthesis is DNA is called replication, and synthesis of identical copies of DNA, it's called replication of DNA, because the strand of DNA are complementary. Both strand carry 8 am energetic information, and it passes or it serves as a templates for the synthesis of complementary new strand, and the new strand which is called daughter strand, in which DNA molecules are identical to the original or parent strand, they contain all the original genetic information. So, today in this lecture we had discussed about the chemistry of DNA and how the nucleotides join together with other nucleotides. The phosphate group of one nucleotide joined through the phosphoester bond with other nucleotide, while in DNA the OH group which is present at the 3rd carbon atom act as a nucleophile, and that nucleophile attack to the other phosphate group to another nucleotide and make a bond, covalent bond, and that make DNA molecule or double helical structure very strong and stable. Thank you.